Okay, so we have our confidence cell prep. It's ready to go. And now all we got to do is take our DNA and aliquot it into the Petri strip, or rather the PCR strip, in order to get it good to go. So I've done the first few. I'm just going to do the last couple. So I'm going to take 1 microliter, and I'm going to just aliquot it out. So here's my DNA. I'm just going to put it right there. 1, 2, 3. Now, I don't know if you look closely, so I've actually done it to all of them. There's a little, here, if I can point it with a point, maybe. There's a little bit of a bead of liquid on the left-hand side of, so, right there. If I touch my pipette tip and move it around a little bit, right there. So all of the PCR tubes in that strip have one of those little liquid bubbles. And so that's our DNA. So we have DNA in that tube, and all we're going to be doing is adding 100 microliters of our cells to those tubes. So 100 microliters. So what's going on here is I have one thing of DNA, and I'm transforming 9 different cell lines with this one piece of DNA. So this is kind of the opposite of what usually happens, where you have one cell line, and then you might transform in like 5 different pieces of DNA. So we have 9 cell lines, 1 DNA. So I took the same piece of DNA and I aliquoted it all out of those Petri strips. And now I'm taking my 9 different cell lines, and cell line 1's going there, cell line 2 is going there, cell line 3 is going there, cell line 9 is going there. So without further ado, I'm going to open up my cell prep. And 100 microliters is going into that DNA. Now I have this aluminum block down. I've added some water to make sure that there's contact between the tube and the block, and that block is on ice, so this entire thing is kept cold. Everything is chilled. It's very important whenever you're doing any sort of transformation. You need to keep your cells cool until you do the heat shock. So here comes 100 microliters. And I'm actually going to try to dispense my cells right on that bead of DNA that I put down, so it'll kind of mix into it. There's 1. Here's 2. Again, I'm aliquoting the competent cells right on that bead of DNA, so that'll save me from having to resuspend and mix with a pipette tip. And since I'm using all of these different cell lines, I need to change pipette tips each time. Again, I'm doing this all with a flame. Sterile technique is important here. You don't want to get any sort of a contamination. Sometimes you can't help it, but you got to do everything you can in order to minimize contamination. So there's number 4, 5 more. Like any sort of medium or high throughput, you always need to pay attention to what well you're on. So you need to have some sort of a mental count going on. So now I'm on 6, because it's very easy to get confused. Even if you catch yourself, you might catch yourself too late. Like you might double dip something and accidentally contaminate, which is no good. That will ruin your experiment. So just 3 more. Oh, see, now I'm confused. 2 more now. And last one. So now we have all of our tubes have cells and DNA, so I'm going to cap that one. I had 9 samples, so I couldn't fit them all in the Petri strip. And I'm going to take the Petri strip tops and close it up. Oops. Doesn't fit. What I like to do is I like to cap the far left and the far right, just to make sure everything's down, and then I just kind of work my way across, just so that nothing sneaks inside. Uh-oh. Just had a capping failure here. There we go. So what happened there, which is actually kind of common, is that if this is kind of a blown up version of our tube, and the cap is supposed to fit in like that, the cap kind of went like this, and it got a little bit smushed, so I just lifted it up and then forced it back in. So I don't know if that representation made sense, but hopefully it did. So now all of our DNA is mixed with our cells. I'm actually doing a couple of other transformations here off to the side, so you can ignore those. We'll just pretend those are other competent cell preps. But now we're just going to wait for a few minutes. The standard is 10 minutes, so we can get away with 3 minutes. So I'm going to set a timer. And after about 3 minutes, we're going to heat shock this. But in the meantime, let's review. And before we review, I just want to say a couple more things. That is that I put in plasma DNA here, which is 1 microliter, but you can also transform in ligations or whatever you want. Probably wouldn't work so well for I don't know, like a Dutsenko Wanner or something like that. You would want to do an electroporation. This isn't the most efficient protocol, but it's pretty good, it's easy, it's pretty good for what we do. So, quick review. We started off with some overnight in these 9 blocks. We didn't film that, but all I did was I took 3 milliliters of LB and aliquoted them into these empty wells of our 24-well block, so 3 mils. And then I did a 1 to 25 dilution, which I think came out to, I think it was 120 or 180 microliters. So I took 150 microliters, because I can't remember what it was, from our overnight saturated culture, and put it into our fresh 3 mils of LB. And so I did that for each one of my cell lines, so I had 9 dilutions. And then I put that shaking at 37 degrees for about an hour. And after an hour I looked, it was a little bit cloudy, and I put it immediately on ice, swished it around. You want to swish it around, that's important, because if you don't swish it around, then the bottom of the culture will get cold, and the top may stay warm. So you want to swish it around, make sure you get a good contact between your 24-well block and the ice bath. And then after it chilled, we spun it down, we emptied all the liquid, we tapped it down to make sure we didn't have any excess liquid, and then we resuspended in 100 microliters of TSS and then 10 microliters of KCM. When we add the TSS, we can make negative 80 stocks there if we want. Generally, you're not going to be doing that. You're going to be doing a large-scale competent cell prep if you're going to be wanting to make negative 80 stocks. So we just went ahead and added in the 10 microliters of KCM. And then after we added that in, we vortexed it, and then we let it chill on ice. While it was chilling on ice, we added our DNA to the Petri strips, and after that we just took our competent cell prep and we aliquoted it into our Petri strips, which already had the DNA in there, and we ejected the cells onto the DNA dot, so it kind of mixed it for us. And then now we're just going to chill it on ice. We're allowing all of the DNA to hopefully touch our cells and getting prepped for the actual heat shock. We're going to heat shock, we're going to recover, and then we're going to plate. Don't know if we're going to film that, we've already done that a few times, but that's kind of the entire process. Another note was that I'm doing 9 cell lines and 1 piece of DNA. That's not always how you're going to be doing it. Oftentimes you're going to be doing 1 cell line and then 9 pieces of DNA, so just keep that in mind. If you're going to be doing a lot of cells, you can use this 24-well block technique. If you're not using the 24-well block technique, you would be taking, if this is your overnight and you want to do a competent cell prep with this, or actually maybe this is a better example. If this is your overnight and you want to make a competent cell prep of this, you're going to do a 1 to 25 dilution in another tube. So take a fresh tube, fresh LB, 1 to 25 into here, grow that up for an hour, take out 2 mils, put it into a 2 mil epi tube like this, spin it down in your centrifuge, bring it, after you empty it, then you can bring it back up in 100 microliters of TSS, 10 microliters of KCM, and then it's the exact same procedure from there. So that is small scale comp cell prep. Let's cut it here for a second for the heat shock, and then we'll be right back.